So welcome everyone to session 4 of the 6th Cancer Cachexia Conference. Thank you for joining us today. We hope you all enjoyed the interactive poster viewing and had a chance to meet with some of the various authors. My name is Rishi Jain. I'm a GI oncologist at Fox Chase Cancer Center in Philadelphia, and I also do research in nutrition and cancer cachexia, and it's really a pleasure to be here with you today. We will be moderating a session on nutrition imbalance and strategies in cancer cachexia. And I would like to introduce my co-chair today, Dr. Trude Balstead. So I'll let her take it away.
Thank you. Thanks to the organizer for inviting me to moderate this session together with Rishi. It's a pleasure to be here. And before we start with the first speaker, some housekeeping notes. The talks are being recorded, as you probably know, and they will be available on the platform for WUHA for the next days. So please use the Q&A box on the bottom if you have questions to the speakers, and use the chat function if you need administration help during this session. And now let's get started. It's a pleasure for me to welcome our first speaker, Dr. Klaske van Noren from the Wageningen University in the Netherlands. And she will discuss the gut-brain axis in the cancer cachexia-related anorexia cachexia syndrome. So Dr. van Noren, please.
We can't hear you, Dr. van Noren. Oh, you're on mute, Dr. van Noren. I think you're okay now.
Yeah,
yes.
Okay, thank you. Sorry for that. Hello, my name is Klaske van Noren, and I would like to thank you organizers for inviting me to this exciting conference. And I would like to start with saying that I would like to give a presentation on really hypothesis building on how gut and brain axis could be involved in anorexia cachexia syndrome, and the research that we did in connection to this, and how it could relate to maybe new angles to look into the cachexia syndrome. And as has been said by many different speakers now, many different mechanisms are involved, and probably the solution is not in applying to one mechanism, but maybe specifically targeted on the patients, look into what is the background of the cause of the cachexia of these patients. And so what we have been looking into are the hypothalamus, the gut and the muscle, together with the tumor, and how the crosstalk is going on. And of course, if I say we, I really mean we, because all the PhD students involved have really carried out a lot of the work, to which I come later. So I have to get my next slide. Yes, there it is. So if we look into the gastrointestinal tract and involvement of the gastrointestinal tract, people often think of the gut microbiota and how they influence the homeostasis. And there has been a lot of literature on the gut-brain axis and the involvement of microbiota and microbiota changes. But what can be realized is that the gut is also quite a more complex organ system, and that, for example, 70 to 60% of the immune cells are in the gut, which means that if you talk inflammation, immune cells can really contribute. Next to that, mucus is quite a complex system, and that also can really be regulated. And since we are in the nutritional session, it's also becoming more clear that also mucus might be being influenced by nutrition. And of course, immune cells of the gut, maybe with nutrition, the gut is the first that needs nutrition. So maybe it's also a target for nutrition. And another thing are the many neurons that are present in this system. And if you look into the connections to the nervus vagus, to the hypothalamus, it's a very complex system how it interacts. Of course, there are the bacteria, and they will be of high influence. And then there is the microbiome produced by the bacteria that also has its influence on the complete system. So what could be the influence of the intestine in cancer? Well, there are different reports on intestinal permeability in cancer, and also Laura Bindels has done nice work in this field. And for example, there is this study from Jiang et al. Who shows in gastric cancer patients that there is a difference between cachexia and non-cachexia patients in intestinal permeability. And intestinal permeability might cause the bacteria from the gut or bacterial compounds like LPS, but also like compounds from the gram-positive bacteria to come into the system and trigger an inflammation together with the immune cells that are already there. And like I said, there is also the mucus of the intestine. And if we look into the mucus, for example, in the colon, you have to realize that it's really nicely organized. So you have a firm layer that is attached directly to the epithelial layer. And this layer should not contain bacteria, but it's the loose layer at the top that contains the bacteria. And so, for example, this protein MUC2, it really contributes to the network of the mucus layer and to this firm attachment of the mucus layer. And so mucin 2, which is produced also by the goblet cells, it builds this mucus layer. And next to that, of course, this is the layer where the IgA can be found, which is also part of the defense system. And one of the hypotheses of a decrease in epithelial integrity also consists a decrease in mucus, which is here very schematically reproduced. And what you see is that if the mucus fails, that bacteria can enter. And of course, it's not this black and white, but it's just for giving you a kind of imagination of it. And so, if we look in mucus and aging, and it has been discussed before, cancer and cachexia often occurs as an old age. And what you see is that mucus thickness, if you look at young and elderly people, but that it might decrease. But what you have to keep in mind that most of those data are from animal studies. So that indeed the mucus is changing from young to old. But what the idea is that it can be translated also to the human situation. And if you look also into how much goblet cells produce mucus, so how many goblet cells are there is less in the old compared to the young. And if we then look into the cancer literature, which is also more in mice than in humans, then you see that MUC2, which is this compound which makes a tight layer for the mucus, is connected to the development of colorectal cancer and to colitis. So both the inflammation, but also the cancer forming part. And so what can be hypothesized is that in colorectal cancer, you might have this reduced MUC2, which might also contribute to an increased intestinal permeability, which might in the end also contribute to cachexia. So what are the ideas of how there is crosstalk between the microbiota, between intestinal permeability and the brain? Well, if you look at the intestinal microbiota, there are compounds which come from these microbiota, like LPS, if it's gram-negative bacteria. And if tight junctions are loosening, then these kind of compounds might come through. And this means that inflammation can occur. And then via the vagus nervus, it might be transmitted to, the signals might be transmitted to the brain, or it might go via the plasma. And next to that, you have all the communication between the bacterial compounds and the gut-brain barrier, both via the vagus nervus and via the blood-brain barrier, the circulation. So if we look into literature that is studying the gut-brain barrier, then and this intestinal permeability, then the idea is that this intestinal permeability leads to activation of the HPA axis. The hypothalamus is activated, and due to that, cortisol is formed, and cortisol might further increase the situation, so amplify the situation, and in this way it amplifies the intestinal permeability. But you might also be aware of the studies from Daniel Marks, in which he showed that the hypothalamus and the HPA axis and cortisol is also involved in reducing the muscle mass, so in that way involved in cachexia. So it might in this way work in two directions, that cortisol stimulates the breakdown of the muscle, but also stimulates the intestinal permeability. And in that way, you might have a compound that worsens both situations. So if we then go to a scheme based on an earlier presentation, an earlier paper of ours, and which I adapted further, what you might have is intestinal permeability of the gut, leading to bacterial compounds, to inflammation, and also the tumor itself causes inflammation, and this inflammation causes again the intestinal permeability, and together they might work on the cachexia, so the breakdown of the muscle. But next to that, these bacterial compounds are also sensed by the hypothalamus, and the hypothalamus can also sense the inflammation, and in that way amplify the signal, and via cortisol increase the cachexia and increase the intestinal permeability. And moreover, the activation of the hypothalamus increases anorexia and fatigue, which both contribute to an increase of breakdown of muscle. And this is a hypothesis for which we have some background and some data. And I think it's worth to go into finding the translation also to the human situation. So inspired by the findings of Laura Bindels, that gut permeability is involved in the C26 model, and in other models that she showed, we also looked into this intestinal permeability, and then specifically also the connection to the hypothalamus. So these are unpublished data. They have been submitted, but so please treat them also that way. And this is work from Xiaolin Li and from Tosca Holtrop and Fleur Janssen, three excellent people, PhD students and master students. And they together really made very nice work. And if we looked into these animals, these are pancreatic cancer models, so KPC, and also 4662. And this work was done at the lab of Daniel Marks. We had a very nice cooperation with him, and Tosca was at his lab. And what we saw is the classic Cachexia phenomenon, and as has been published, 4662 is less severe in Cachexia, which is also a result from decreased effect of food intake. And if we then look into the intestinal permeability, what you can see is that in this pancreatic model, there's more intestinal permeability than in this 4662 model. Although there is still intestinal permeability, which was even as high as DSS model. And if you then look into the hypothalamic gene expression, what you can see is a very clear inflammatory pattern on many different genes. So apparently they coexist, the inflammatory status of the hypothalamus and the intestinal permeability in the gut. And what we then wanted to know is, can those two compounds have the intestinal permeability and the inflammation in the hypothalamus, can they talk with each other via compounds produced by the tumor, produced by the gut, and can they be amplified? So we first looked at different cell lines. This is an hypothalamic cell line, and this is an myoglial cell line. And what we saw that the hypothalamic cell line, so the neural cell line, was more sensible to LPS when it concerned the production of IL-6 than the microglial cells. And what's intriguing about this is that it might have a kind of signaling function. And next to that, we looked into different animal models, and we made secretomes from the cell lines that were used for these models. So they were cultured in vitro, and then that medium we used to stimulate the hypothalamic cells. And what you can see is that if you have LPS and you have this secretome of the animal of these cells that were used in animals to produce cachexia, that you get an amplified signal. And this is true for the different models like Lewis Long, KPC, and 4662. And what you can see here is that KPC is producing more IL-6 than 4662, which is also less causing less cachexia. So this is nice work from Xiaolin Li. And if we then look into what could cause this, well, we made a kind of a mimic based on inflammatory compounds that we measured. And when we left out PG2 from this mimic, the effect was gone. So what we also did was we used a concentration range of PG2, and then the amount of secretome that contains the same amount, and we mimicked them together, and they really were quite similar, the responses. So it seems that PG2 has a role in the amplification of the LPS signal. To further verify that, we used just single compounds. So LPS, C26, in this case, C26 with LPS, and then the mimic with LPS. And if you then don't add PG2, you see that the effect is gone. So PG2 might play a role here. So prostaglandin 2, E2, and what we then next did is we communicated with Daniel Marks again, and we looked into the PG2 concentrations of animals from a different experiment. And in this case, there's again Cachexia, and also there is a difference in the KPC and the 4662 model, as you can see, both for muscle mass and for total food intake. And what you can see in the plasma PG2 concentration is that KPC seems to have a higher PG2 concentration than 4662. And there is a difference, a significant difference if you make it into a log transformation, because there's quite some variation between the different animals if you just look into it. So it's not normally distributed. But this is another hinge that it might be that the prostaglandin system, together with the LPS sensing, might amplify a signal in the hypothalamus. So then we have this hypothesis that this kind of signals might amplify each other, and then what could be done with nutrition, because that's or lifestyle, because that's what we would like to aim at. And students of my lab, they made a nice review, and what they found is that there is some indication that lactobacillus might have, if you look into the animal models, might have an effect on muscle. So, and then specifically for lactobacillus, there is some data available. And I say some because these are animal models and they are not a very large number of animals. And so there are no human data yet. And the question is it really an effect of lactobacillus? Because there's no, we found no other bacteria that were, so probiotics that were tried, except for bifidobacterium, which had a similar kind of effect. So, but if you look into literature on aging, you also find a similar effect. So it might be that with probiotics, you might have some positive effect, but what we have to realize is that often effects in animals, because they are all the same age, they are all treated the same, etc., same kind of tumor, that the effects are far more neat than they ever will be in humans. So you might use it as part of a therapy, but it will never be completely the bullets you need. So what is also something we should think about when talking about nutrition and probiotics is that these bacteria produce a lot of very beneficial compounds, so the microbiome, and maybe we can directly stir the right...
I'm sorry to interrupt you, Van Noren. Your time is soon up.
Okay, thank you.
Yeah.
Yeah, that's good. Thank you. So the microbiome might contribute, and we have to look into that. And next to that, the nutritional status, what we found is that if you look into intestinal permeability, that if you measure it into an in vitro system, that it really matters if the intestine is fat or not. And next to that, the mucus can be influenced by, for example, oatmeals. And in that way, you can see that if you consume an oatmeal, it can protect against infiltration of small compounds better than a normal meal. Next to that, what we have found is that with exercise has been known to can be causing intestinal permeability. But if you retrain so that you give the same exercise repeatedly, what you see is that this effect changes, and that maybe exercise can also induce GLP-2, and GLP-2 is a compound that heals gut. So in that way, you might also use exercise in a way to heal the gut. And that's, I think, another way to look at exercise. And another way to get this hormone up might be, these are studies we did with tube feeding. And what you see is if you give it on the stomach or the jejunum, that it does not really matter for protein or glucose, but it matters for gut hormones. And for GLP-2, for example, if you give it in the intestine post-rites, you get more GLP-2 than if you give it on the stomach. So you might also heal the gut by changing the way you give your nutrition. So, and the last part is that dehydration also influence to a high extent how the gut is influenced by intestinal permeability triggers. So, to conclude, I would like to give you the thought that the HPA axis is involved in cachexia. I think Daniel Mars and our group already have some nice data for that, that the gut might be involved in it, and that if we want to look at the gut, we should look broader than gut microbiota, but look also at mucus, look at energy status, at hydration grades, at the microbiome, how the immune system is evolved and how it can be triggered and be involved in the intestinal permeability, how we can use exercise to both give treatment for the muscle and for the gut, and in that way maybe trigger GLP-2. And these are all the fantastic people that worked on it. And I also would like to thank Daniel Mark's lab for it. And thank you.
Thank you so much for a wonderful talk, Dr. Van Noren. So sorry for interrupting you.
No,
we have time for a couple of questions, and please enter your questions into the Q&A, and we already have some. This first question is from Dr. Michael Ramage. Hi, Klaske, very interesting talk. There were a lot of mentions of mouse models and hypothesis translation. Have you looked into testing some of these theories in human cancer, non-cancer resection specimens?
No, not yet. We would like to do that. So I think we should move to translation, and I think we have now some data to support that we can move to that stage. Yeah.
The next one is from Dr. Dan Runko. Thank you for the great presentation. Does the increase in gut permeability increase the risk of infection in mice with cancer or immunocompromised? It's a theoretical risk with myeloma suppressed patients, which makes some clinicians hesitating to add probiotics, but not sure if there is any negative side effects in animal models. Thank you.
Yeah, Yeah, I think there has, of course, been a trial in pancreatitis, which went quite wrong with probiotics. And I think you have to be careful to, for example, giving it past the gut, and that you make sure that the gut, that you have the right dose, so that if the gut normally a large amount of the bacteria dies in the gut because of dies in the stomach because of the low pH, and so you should be sure that the amount of probiotics that you give is also according to the status of the patient. And if you are thinking about too vulnerable patients, then maybe we should go to prebiotics and say what kind of fibers do we need to get this good homeostasis of bacteria ready. So I think there are more ways to go there, and also what kind of microbiome is produced by this bacteria that we need. So and that's also why I said we should move further than only probiotics. So
thank you. We can have room for one more question from Dr. Kanya Mela. Very exciting talk. Did you also look into impact of chemotherapies on gut, hypothalamus axis, and cachexia in your models? I wonder how restoration in chemotherapy treated patients with probiotics will cure cachexia phenotype.
Yeah, that's a very interesting one, and I don't know the effect yet. I know that, for example, for cisplatin, that you have very tremendous effects on the gut, and so also in that situation you will have intestinal permeability, definitely, but we have to look into it before I can say that it's there. So
thank you. I think we have to close your talk now, yeah. Dr. Van Noren. Thank you very much.
Okay.
So thank you, Trude. I'd like to introduce the next speaker. I'd like to introduce Dr. Cristina Sintra Gomez Marcondes from the University of Campinas, Brazil. She'll be talking about nutrition as a coadjuvant therapy in cachexia preclinical models.
Right.
I think you just have to change it into the presenter.
Yeah. Is it okay?
Looks
yep.
Good.
I'd like to thank to all organizers to invite me to be here. It's a pleasure for me, and I would like to show and share with you some results of nutritional approach and preclinical model. Some studies have shown that when cachexia starts, the most challenge for this process is to stop the progression and also treat the severe clinical condition in these patients. Most of these studies have shown many strategies of coadjuvant treatment, such as nutritional approach, in association to conventional clinical treatment. So we have recently written a chapter about probiotics, amino acids, antioxidants, and polyunsaturated fatty acids, which are very common nutraceutical used, generally used as a coadjuvant, and QKIT showed some actions for these, for example, reducing anorexia and restoring or minimize the changes in most of physiological functions, and mainly reducing the tumor cells activity. So I'd like to show you some few of our results about leucine supplementation as a nutraceutical point that act signaling, acting as a cell signaling improve the host tissues and organ, and increase protein synthesis and decreasing protein degradation. And we can show these results in a model of cachexia. It is the Walker 256 tumor model that has present a very closer points to the clinical feature, such as anorexia, body weight loss, and especially adipocyte decrease, and especially the skeletal muscle associated with very alterations in lipid and carbohydrate protein metabolism. And also recently we can see changes in cardiac muscle, and also an upregulation of the ubiquitin proteasome system and affecting the mitochondrial activity. So here is our hypothesis that how can leucine interfere with tumor cells, and if the tumor mass increase with this supplementation, and the carbohydrate metabolism is entirely affected during cachexia. So how can leucine modulate this and improve this metabolism? And also talking about the protein metabolism, as we can see that the weight loss is such a problem for these patients, for these cachectic patients. So how can leucine improve protein metabolism and also increase the lean body mass for these patients? And our last question is to prevent the start of cachexia. It is a very intriguing question, and we try to improve the host responses and by epigenetic modulation, so we can feed the mothers with a leucine-rich diet and see if there is some results in cachexia evolution. Our first question is, how can leucine affect tumor cells? And here I can show you our experimental model that we distribute Wistar rats into 4 groups, control and tumor-bearing groups, and 2 other groups fed 3% leucine-rich diet. And here Dr. Viana and colleagues showed that in a PET scan assay that there is a decrease in glucose uptake by tumor cells. And these results show no increase and no change in tumor mass. So here another point, that the general process of glycolysis to lactate and low ATP production by tumor cells are diverted to oxidation reduction process, showing a shift in metabolism which altered the tumor activity and not favoring formation of new metastasis sites under leucine supplementation. So here, talking about the carbohydrate metabolism, how can leucine affect this metabolism? So here we can see that the liver glycogen is deeply depleted and restored. Sorry, a little bit. And restored by leucine-rich diet. And also Dr. Viana showed that the main enzymes and related to waste process are modulated by leucine supplementation, reducing the gluconeogenesis process, reducing the chlorine cycle and restore the energy demand for these animals. Also talking about muscle, and we can see that the glycogen is deeply decreased and restored a similar value of the control group. And also the glucose transporter and exokinase and lactate dehydrogenase and pyruvate dehydrogenase are modulated by leucine-rich diet, which conduct to a better muscle response related to the improvement of energy supply. In a in vitro experimental model, we can see that we worked with C2C12 myotubes and leucine supplementation, and we mimicked the tumor effects. And here in metabolomic assay, Dr. Cruz showed that the metabolites in these cells conducted to energy generation. And also he pointed out that the main cellular component affected by leucine is mitochondria, improve this activity. Here in this electronic microscopy, we can see an increased biogenesis of mitochondria in this leucine-treated group. And also here in a proteomic assay, we can see that the enzymes are actually modulated by leucine supplementation, such as a glycogen phosphorylate that is maintained in leucine group, which reduces the muscle glycogen mobilization. And the pyruvate dehydrogenase is restored in leucine group, where we have more pyruvate conducted to 3-carboxylic acid process, and also an increase in ATP synthesis, showing that the leucine increase the availability of ATP production in this muscle rather than just lactate and less energy supply. Here, Dr. Viana also highlighted that OXPHOS enzymes are improved in this muscle of these animals treated with leucine-rich diet, increasing or improving the muscle energy supply. Here are our third question or third hypothesis: how can leucine improve the protein metabolism and increase the lean body mass of this host? So we can see here the increase of the methyl-histidine, that is the metabolite of muscle waste. And here we highlighted the 10th day of tumor evolution, showing the start of cachexia. So here, if we can improve the process, increase the protein synthesis, and also having a positive point of the protein turnover, we can see that the muscle mass can be preserved or minimize the effect of cachexia process. Here we can see the main muscle protein, the myosin heavy chain, here preserved by leucine-rich diet. And also here is these other graphics show the maintaining the activity of this muscle tissue, increase or maintaining the incorporation of leucine, and also the incorporation of its main metabolite, that is the ketoisocaprate caproic acid. Here, in another study with Dr. Chiocchetti, we can see that the cardiac muscle decreased during cachexia process, even with leucine supplementation. And despite this reduction in heart weight, we can see a decrease in protein degradation. Probably is an adaptation of this tissue maintain the main activity, and also increase in protein synthesis being similar to the control, and also driven to positive protein turnover, minimizing the damage caused by cancer cachexia. Also here with Also here with in a work with Dr. Tonedo, we can see the cardiac biomarkers that are modulated by leucine-rich diet, and also minimize the pro-inflammatory process and decreasing the ischemic risk driven by cachexia process. Another important point is the maintenance of the insulin level, and also decreasing the pro-inflammatory cytokines, restoring to the similar value to the control group, and also minimizing the proteolysis process. So here, Dr. Viana showed the down-regulation of the ubiquitin proteasome in these animals treated with leucine-rich diet, and regulating this proteolysis process, and not only the ubiquitin subunits of ubiquitin proteasome system, but also the promoter here, FOXO and MRF, are modulated by leucine supplementation, also decreasing the proteolysis process. But not only the molecular assay, but we wanted to see and to analyze the muscle function. Here are 3 tests that we can see: walking test and behavior test, and muscle strength as a muscle function analysis. And here we can see the footprint before the tumor implant and after tumor implant, and we can measure this footprint intensity. And here we can see the maximum contact area, and we have a slightly decrease, but not so intense as the tumor bearing without nutritional supplementation. And not only this time moving, but the distance and the speed of these animals maintain similar to the start point compared to, compared to the tumor bearing animals without any supplementation with a control diet. So the muscle strength here, we have a slightly decrease, but it is similar to the start point and different from the tumor bearing without supplementation. So we can see that diet rich in leucine can improve the muscle function, reducing the decline of walking and behavior, and also the muscle strength in these animals that are affected by cachexia. And our last question, and how we can prevent the cachexia, and using an epigenetic modulation. And here are our experimental protocol, and we feed these mothers with control diet and with leucine-rich diet during pregnancy and lactation. And these offspring from these mothers are growing up until adulthood with control diet. And also when they are adult offsprings, they are distributed into these 4 groups. And here we can see that Dr. Miyaguchi showed here a slightly decrease in Cachexia index, and also maintenance of the hepatic activity, and also increasing the anti-inflammatory response in this liver. And here she showed here an increase of survival curve for these animals. So decreasing the anorexia process is a very interesting point and a positive effect of maternal diet. Here we can see the decrease of food intake, but not so higher and different from the tumor bearing without any supplementation. And she also found muscle maintenance because we have a decrease of protein degradation conducive to a positive protein turnover in these muscles. And in a metabolomic assay, we can see that the metabolites here conduct to alter metabolic and impacted metabolic pathways that alter the muscle response, conduct to an anabolic process, and despite to have some metabolites that it's not found in a common protocol, but we can in general see the positive effect of maternal diet over the cachexia damage process, increase the host adult offspring responses. So in conclusion, and for future analysis and maybe translational process, we can see that the nutritional supplementation could be an interesting approach in preclinical models. And leucine can modulate the affected function of the skeletal muscle, heart, and liver, improve this activity. And also epigenetic effect by the maternal diet, fed leucine-rich diet can preserve and minimize the damage effects driven by cachexia. And further studies are undergoing in our laboratory for future answers. How can leucine counteract and drive to better condition? So I would like to thank to all my team, and here are a few of us here, and it is a pleasure to be here with all of you, and I am pleased to answer any question. Thank you.
Thank you so much. So we do have some questions coming in. That was a great presentation. So this is from Pankaj Singh. Thank you for the great presentation. Leucine induces mTOR that can enhance glycolysis. The lack of aggressiveness in tumors in mice on leucine-rich diet is intriguing. Have you tested the effects of leucine-rich diet on other cancer cell line models to test if the reported effects are not tumor cell type specific?
Thank you for this question. And we have results published that the emitter is increased in tumor cells under leucine supplementation. But as we showed here, that the less glucose uptake, it is not increasing the tumor cell proliferation. We are doing in other cells, such as C26 and also Lewis carcinoma, and to see if there is some point that can driven us to another experiment or to another answer for this. But it is, we have done these results and published yet, okay? Thank you.
So we actually have a follow-up question from, so did you measure if the leucine-rich diet increases circulating leucine levels?
Yep, yeah, it is increased, and also we have a better absorption by the gut, leucine and isoleucine and valine. It is improved by this diet rich in leucine. And also we have an increase in serum of these amino acids, especially the branched chain amino acids.
Okay. And then are the improvements in cachexia, are they primarily due to increased leucine in circulation or due to changes in the gut microbiome? Do you have any data regarding that?
Increased leucine circulation and chain. Oh my God, I don't know if we have the 2 points increasing here, the leucine increase in circulation, and also improve the gut microbiome as well. I think both potentially this answer for us, this point, this result for us, okay?
Great. Next question is, have you tested leucine and glutamine associated supplementation or another association?
We have done another experiment, and I showed the most recent experiment for this conference, but we have tested the leucine with others and also glutamine, and it has an interesting point, but most of the responses are equal or similar to the leucine rich diet. I would like all these other amino acids improve and potentialize the leucine response, but leucine is the first one.
Great. Well, we hope to see some of that data in future conferences. So another question, great talk. Since your studies have shown effects in mitochondria, have you looked into autophagic markers in your model?
Not yet. Autophagy, we can see, we trying to see this. Leucine can minimize the autophagy, but we haven't done this yet in our studies.
Okay.
But in C2, C12 cells, we have done only analyze the leucine in C2, C12 myotubes to see how the effect of leucine, and we can see that it is decrease the autophagy. But in animal model, we haven't done it yet.
Okay, we'll have one more question. So any effect of leucine on kidney function that you've observed?
Sorry, can you repeat? Sorry, please.
Was there any effect of the leucine on kidney function in any of your studies?
We have done many years ago how to balance and how to achieve the right percentage of leucine. And when we increase much more amino acids or increase the protein, we have some kidney function affected. But in this concentration or in this percentage, no, we haven't seen any alteration in kidney function.
Great. And actually, I'd like to go back to one more question that I think I missed. This question kind of has to do with how to translate this data to humans. So the question is, thank you, very interesting talk. Leucine supplementation has been tested quite comprehensively in clinical trials of sarcopenia with effects on muscle protein turnover and maintenance of lean mass being mostly small or even absent. How do you expect similar challenges in translating your results to scenarios of cancer cachexia patients?
As I pointed out in the last part of my talk and in conclusion, we are now going to make some collaboration to translate for this translational approach and to see, especially for colon cancer and lung cancer, that there is a high index of cachexia, and we can see and see if we can work with a similar process and see if we can translate, do a translational process for the clinic. And now we have done some collaboration and we are starting for this, not yet, no.
Well, great. Thank you so much for the excellent talk. And I'm going to hand it back over to my co-chair.
Thank you. It's my pleasure.
Thank you, Rishi. Now we move on to the short talk part of this session. We have two speakers. They have submitted abstracts and they have 10 minutes and there is time for some questions. So again, use the Q&A and address the researchers. So it's an honor for me to introduce the first speaker, Dr. Tor Erik Russen from the University of Oslo, and he will talk about autophagy in flies.
So do you hear me clearly? Yes, probably. Okay, I would like to start by thanking the organizer for the invitation. It's a huge honor for me to come here and talk about our work. So we'll talk about autophagy, we'll talk about organ wasting, and we'll talk about that in the context of tumor growth in flies. And probably the first question that comes to your mind is, why the fly? Why would you even think about using the fly to answer these type of questions? The answer is that flies has been remarkably successful in unraveling basic cellular mechanisms and genetic mechanisms of cell biology and even disease mechanisms for more than 100 years. And flies like us has a bilateral brain. It has an alimentary canal with stem cells that replenish the gut lining. It has a kidney called the Malpighian tubules. It has this female fly has ovaries. It has adipose and liver tissue in the form of the fat body. It has muscles. It has a dorsal open heart that keeps the circulation going. It has patrolling immune cells which detect invading bacteria and even parasites. and can detect and purge tumor initiating cells. And most importantly for this talk, we can genetically seed labeled tumor initiating cells by taking away tumor suppressors and overexpressing oncogenes under physiological conditions in a manner similar to what can be done in mice. So in this context, we can ask questions relative to tumor host biology. So we seed tumor cells to make carcinomas in this neuroepithelium that is attached to the brain called the ion tunnel disc. This is a simple one-layer epithelium. And these tumor initiating cells grow over time, and this is what you see here as these green clones here. So we can do that by overexpressing oncogenes, overexpressing the RASV12 oncogene will form benign tumors in this ion tunnel disc, as you see here in this confocal image. And on the upper quadrant, you see the genotypes of these different compartments. So this is the transformed compartment with GFP in the confocal image, the neighboring microenvironment, which is wild type in this case, and the rest of the body in this square. But when RAS is combined with a tumor suppressor, these cells behave completely different. They transform, they grow indefinitely, they refuse to die, they express matrix metalloproteases, degrade the basement membrane and invade the brain, and over time and eventually kill the animal. And different complex traits of this tumor model can be studied using even the fly. So we'll talk about autophagy. I guess you're familiar with autophagy from other studies, but just to remind ourselves, autophagy is conducted by isolation of the cytosol that can contain organelles like mitochondria, protein aggregates, and more. That is shuttled to the lysosome, where upon fusion, this inner membrane is degraded together with the content, and by enzymatic degradation and low pH, the nutrients in the form of fatty acids, nucleotides, and amino acids are recycled for bioenergetics of the cell or can be shuttled to the rest of the body. So we can follow this process by using markers like labeled ATG8 or comparable to LC3 in mammals. And we can manipulate the process by taking away enzyme complexes required to initiate the process, like ATG13 of the ALK1 complex and ATG14 of the P3 kinase class 3 complex, or using pharmacological agents like SAR405, which is specific for the P3 kinase class 3, or chloroquine. Now, our previous studies studying tumor growth in flies showed that in an inflammation-driven tumor model that is dependent on TNF-alpha signaling and interleukin-6 type signaling for transformation can induce a microenvironmental autophagy stress response that is used to feed the tumor. And similar results was obtained by Alec Kimmelman in mammalian cells and in mice. In these studies, we also discovered that the presence of tumor cells also elicited a systemic host response with autophagy activation in muscle, in gut, and the fat body, which is comparable to both adipose tissue and liver in mammals. And furthermore, when we looked at these animals over time, we discovered that as the tumor grows, these larvae that are originally white due to the adipose tissue seem to become more translucent, as if the fat is melting away. So not only do we think we can study tumor microenvironment interactions in this model, but the question arose, could it be that organ wasting occurs in this model? And could it be that organ wasting is enacted by autophagy? Furthermore, can flies be a good model to study aspects of cancer cachexia, and may organ wasting contribute to tumor growth? And of course, this is in part motivated by studies mainly by Fabio Penna and Paola Costelli and colleagues, and other observation in patients and samples. And also seminal work by the Norbert Perimon lab and the David Bilder lab in flies showed in 2015 that adult tumor models can induce wasting of the ovary through insulin resistance in flies. And we'll come back to that at the end. So to look at this question comprehensively, Roger Kesri, a fabulous PhD student in the lab, decided to tackle this question. And together with Todd Schoborg at the NIH at the time, and now at the University of Wyoming, we conducted a micro-CT imaging of these larvae over time, as we thought that when the tumor was growing, perhaps something happens to other organs. So we have looked at all the organs in the animal, and I'm showing you here the muscles in green and the fat body in blue and the growing tumor in red. And you see on the right-hand side that the tumor grows immensely over a time period of 4 days, from day 6 to day 10. And concomitantly, the muscle systems seem to degrade. And we can quantify this. So if you look at tumor volume over time, the tumor grows more than 10 times in volume. And importantly, the muscle system originally grows to the normal size at day 6 and then degrade over time in a time-dependent manner. And we can also look at this as a single muscle level, and we see the same. But when we look at the fat body, again, similar to the liver and the adipose tissue in mammals, we don't see a shrinkage in volume, but we do see an accumulation of large lipid droplets that we ascribe to a steatosis effect. Furthermore, these animals lose body weight, and also they degrade in motility over time, probably because of degradation of muscle. That's what we think. And gradually they also cease feeding. But it's important to note here that the degradation of muscle tissue initiates before the cessation of feeding. Now, a related paper coming out just 2 weeks ago from the lab of Julia Cordero did a few other experiments that are enlightening. So full starvation of these larvae do not lead to muscle wasting. Furthermore, there is also steatosis in muscle, and making similarly sized tumors by other manners do not cause wasting. So we think it's a qualitative difference in these tumors, and the tumors can induce wasting in this system. So our next question was, is there a role of autophagy in organ wasting in this model? So I'm not showing you the data here, but we see an increase of autophagic flux in these models as wasting ensues. And what I show you here are pictures of tumors here in this row, muscles here in flap preps, and again, the fat body tissue with steatosis. So you see from day 6 to day 8, there is a massive tumor growth, wasting of tumor tissue and steatosis. If you take away autophagy in this whole animal here, you see a reduction of tumor growth, a restoration of muscle, so no wasting and no steatosis. What we've shown earlier is that we can put back autophagy in the tumor and the tumor microenvironment and restore tumor growth to 60%. And in this type of experiment, what we see is that the tumor growth and the muscle do not waste, and there's no fat body steatosis. So what this is showing is that there is a systemic effect of autophagy in the system, and the systemic effect of autophagy is to enact body wasting. And then when taking away autophagy, you have a restoration of body weight of the animals, although not to the full weight, and a restoration of larval crawling ability. So our next question was, what may be the function of autophagy in the system as these organs waste? So when we look at the serum of these animals and do metabolomics, what we observe is that there is a time-dependent and strong release of nutrients into the serum, into the larval hemolymph. And this is true both for carbohydrates. So the blood sugar in flies is trehalose and to some extent glucose that initially drops and then bounces back. Amino acids are mobilized, and most fatty acids are mobilized. And this happens at day 6, even when before the tumor growth grows exponentially. And at day 8, essentially all amino acids are mobilized into circulation. So the idea emerged that perhaps organ wasting in this tumor model contributes to tumor growth, so we wanted to test this idea. So this is not an easy question to answer, because you need to distinguish the carbon biomass originating or nutrients originating from wasting organs versus the diet. So you have to label either the whole animal first, or label the diet, and then ask the question, to what extent do the two contribute to tumor growth? So Petter Holland in the lab joined up with people at the University of Oslo, Anne Hope Jaren and Dillian Hergopian, to develop a completely new protocol to look at this based on the idea of Anne Hope Jaren. So apparently, the C13 content in the atmosphere is stable over the whole planet, but the two types of metabolism and carbon fixation in C3 plants versus C4 plants are different in the amount of C13 they fixate. So plants from the Americas, like corn and sugarcane, incorporate more C13 than European-derived comparable sugar beet and potatoes into their biomass, and this is equal among all the biomolecules in the cells. So we were able to devise experiments where we can first feed animals on the European food and then on the heavy diet, the American C13 heavy diet, or vice versa, and do experiments and calculations of do the diet or the organs contribute to tumor biomass. So Petter went to the supermarket and bought these ingredients, and also made yeast grown on sugarcane as the only carbon source. And then we can actually look at these from single larvae.
Sorry to interrupt, but your time is
single
up.
tumors,
So,
yes. And when we do that, we can calculate how much is deriving from the two different sources. And what you can see here over time is that the amount of carbons from host organs into tumor incorporated increases from host organs over time relative to diet. So I'm ending here just to summarize. What I've shown you is that autophagy mediates muscle wasting and nutrient mobilization that is used for tumor growth. What I also want to point out here is that flies actually shows most of the hallmarks of cancer cachexia, and the host papers from different labs now show that IL-6 signaling, RTK signaling through MAP kinases, and insulin resistance is acting in both larva models and adult models to contribute to organ wasting in these models. So I'll stop here and thank the lab. I pointed out most of the people throughout my talk, and I would like to thank our sponsors as well. Thank you for your attention.
thank you so much. Impressive work. I can't see we have any questions, but we are actually out of time. We're 2 minutes into the next talk. But have you started already with your new experiments that you suggested was the next step?
Yeah, so what we're looking at the moment is, of course, what in our system is inducing wasting and what is inducing autophagy, and looking into the cellular mechanisms of these processes. That would be the next step.
Do you see how much they eat? Do you monitoring their intake? Or can you even?
We monitor food intake. I briefly went through it very fast. They do stop eating after a while. So you have an anorexia response. And in fact, there was a recent study showing the mechanism of anorexia response in response to tumor. A fantastic paper from the ULab that shows that this mechanism is through a relaxin-like molecule that is conserved in mice.
Thank you. We have some questions. I think we can have one from the audience, from Dr. Henning Langer. Very exciting data. Methodological question. How did you get enough tissue out of the small flies to GC IRMS? Did you pool animals?
We did not pool animals. The sensitivity is actually so high that we can analyze individual larvae.
One more from Dr. Teresa Simmers. Fascinating talk and important demonstration of the role of cachexia in fueling tumor growth. Do the nutritional constitutions of YAP-induced non-cachexia tumors differ from those of these cachexia-inducing tumors?
That's a very good question. I mean, we have to compare those. We haven't done that. So if we make YAP-induced tumors in this same tissue, we don't see cachexia.
Thank you. Okay, I think we'll have to stop there. And I thank you so much, Tor Erik.
Thank you.
Great. So we will be moving on to our final speaker. I'd like to introduce Dr. Mark Beltra from the University of Torino in Italy. He's going to be talking about niacin supplement restores NAD+ levels and counteracts muscle wasting in cancer and chemotherapy-driven cachexia.
Thank you for the introduction, and let me start by thanking also the organizers of the meeting for giving me the opportunity to share our most recent data, specifically on the use of niacin to restore NAD levels and counteract muscle wasting in cancer cachexia. So I have no conflicts of interest to declare. And I will start my presentation with a pretty obvious statement, which is basically that cancer cachexia has a devastating effect on the skeletal muscle metabolism. It is widely known that many metabolic pathways are altered in this pathology. You can include protein synthesis, protein degradation, so increased protein degradation, also affections in glycolysis, but also mitochondrial dysfunction. And all those alterations can be seen as a symptom of a cellular energy crisis, which in the end lead to skeletal muscle atrophy and dysfunction. So according consistently with what I just reviewed, the skeletal muscle of a cachectic animal has a different, totally different metabolism profile compared to control healthy animals. What is interesting to see, this is not only exclusive to the skeletal muscle, but also to other organs, such as the liver, which is a very highly metabolically active tissue. And you can see that many metabolites are also altered in cancer cachexia. This is a good opportunity to remember that although the skeletal muscle has a central key role in cancer cachexia, we have to consider this syndrome as a systemic disease. But what I wanted to highlight about the slide is the fact that in both tissues, we have a reduction in the levels of NAD, whether it is reduced or oxidated. And NAD, as you may know, it is a coenzyme or cofactor, which is involved in many metabolic reactions, but it is essential, particularly for proper muscle mitochondrial function. And that means of seeing that this deficiency on NAD may be one of maybe a contributor to faulty energy metabolism observed in cancer cachexia, not only in the skeletal muscle, but also in a systemic level. Consistently with these results, it has, we around 1 year ago, we published together with Juha a paper in which we reported that the genes involved in NAD biosynthesis were altered. In particular, we showed a huge downregulation of the NRK2 gene, which on the other side, in the healthy animals, is the most expressed one of the biosynthetic pathway. We have also obtained further data on this regard, in which also the KPC model, the KPC animals also show a reduction on the NRK2 expression in the skeletal muscle. And more interestingly, this gene is also downregulated in cancer patients, in the skeletal muscle of cancer patients. And this downregulation is further exacerbated when these patients are classified as cachectic. Therefore, we can say that we can conclude that NRK2 repression, it is a common feature in many cancer cachexia situations, situations, both in a preclinical model, but also in the human situation. So the hypothesis was pretty simple. We have repression of the NRK2 gene, which means that the skeletal muscle cannot utilize the nicotinamide riboside as a precursor to create unsynthesized NAD. And that may contribute to the decreased NAD availability in the skeletal muscle. So the idea was to try to increase NAD the levels by pushing another side of the biosynthetic pathway by supplementing with niacin, which can be metabolized to create a new NADA without the need to pass through the NRK2 enzyme. So basically, the experimental model was pretty simple also, which we used the C26 tumor model in combination with FOLFOX chemotherapy. And those animals were treated daily with a dose of niacin, a pretty high dose in this case. And the first observation was that those animals were losing less body weight in comparison to non-treated human-bearing animals. And similarly, and most interestingly, muscle wasting was partially reduced in both tibialis and gastrocnemius muscles. This is important to say that that was not due to a reduced tumor burden. As you can see, the improvement in muscle wasting was not due to a lower tumor mass. And again, well, similar to the previous results, we also observed an improvement in muscle grasping strength, so in muscle function in the group that was treated with niacin in comparison to the tumor-bearing animals without the treatment. So those promising, we were wondering if those promising results in muscle wasting and in muscle function were due to a restore in the NAD levels that we were expecting by delivering niacin. And indeed, we were able to obtain a recovery on the NAD levels on the treated group. This was not, however, followed by the increase on NADH. But what was more striking was the fact that niacin supplementation had a dramatic impact on NAD levels, but also NADH levels in the liver. And that, again, shows that niacin is not, the effect of niacin is not only directed or exclusive to the skeletal muscle, but it has more a systemic-like effect in different tissues, and probably in other that we didn't analyze. So the first thing that we wanted to understand is which were the effects in the other metabolic pathways of improving or increasing the amount of available NAD. And what we first saw was that partial recorrection on protein metabolism in CYP26 bearing animals. You can see that protein synthesis was partially rescued in the treated group, and similarly, a slight but significant decrease of a significant decrease of the lipidated LC3B protein was observed in the skeletal in the treated group, suggesting lower activation of the autophagy. What was more interesting, however, was to see that the mitochondrial metabolism was clearly corrected and improved by NA supplementation. In these panels here, you can see that GC1 alpha, which is a protein involved in mitochondrial biogenesis, was increased in niacin-treated animals. And that also ACHA and cytochrome C, which are proteins used as markers of mitochondrial mass, were also improved. Additionally, we also were able to obtain a rescue in a mitochondrial respiration, a partial rescue in this case. And all this data together suggest that niacin was able to improve mitochondrial homeostasis and function in the skeletal muscle. But we also were very intrigued by what would be the effects on the metabolism in the liver, since we saw a dramatic effect in NAD levels of niacin supplementation. And what was pretty striking to us was that while CYP26 bearing animals were showing increased protein biosynthesis, this was not corrected by niacin supplementation, in opposition to what we just observed in the skeletal muscle, in which niacin does correct or does change protein metabolism in the muscle. It did, however, improve muscle markers of mitochondrial mass, so suggesting that also niacin is also able to improve mitochondrial homeostasis in the liver. We still, however, lacking some more functional data on this respect, such as mitochondrial respirometry. And finally, I wanted to show you, so the idea behind this slide is that we wanted to further validate the results that we obtained on the CYP26 model. And we wanted to do that by using a model that could more closely correlate or relate to the human situation of cancer cachexia. So to do that, we made use of this animal here, which is the billing-free MSH2 flox mouse. Those animals basically accumulate spontaneous intestinal tumors. Basically, the MSH2 is the knockout of MSH2 is produced only on the enterocytes of the intestinal mucosa. What we cannot see is that those animals do show muscle wasting, have reduced muscle weight. And interestingly and fortunately for us, the supplementation for 28 days of niacin was sufficient to partially or even fully reverse muscle wasting on those animals, then thus supporting the results obtained on the CT6 model. So to conclude, we can say that a niacin supplementation is able to increase NAD availability both in the skeletal muscle and also in the liver, and that this leads to a beneficial effect in muscle wasting. It is able to partially stop muscle wasting in 2 different models of cancer cachexia. And also it is able to improve mitochondrial metabolism, at least in the skeletal muscle and potentially also in the liver. So we propose niacin as a promising anti-cachexia drug by supporting whole body energy metabolism. We foresee its prospective potential to use it in clinical practice. So last but not least, I would like to thank all the people who contributed to this work, specifically in particular, A and Nora from the University of Helsinki that produced the data on the NAD levels and also on the mitochondrial respiration. Paolo Porporato that gave us the samples of the KPC animals, Juha, which is one of the main brains behind the idea of utilizing niacin, and Roberta and Marco for giving us the samples in which we, the human samples in which we assess the NRK2 expression. Finally, I also want to give thanks to the organization that support our research. And I would like to thank you for listening and for staying until the last talk today.
Thank you, Dr. Beltra. So just a couple minutes for questions. So the first question was, did you test for requirement of NAD or involvement of the niacin membrane receptor GPR109 in your phenotypes?
No, we did not test about this. No, we did not. I cannot.
Okay. Okay, couple more quick ones. So did you observe any changes in sirtuin activity by NA supplementation in the muscles? Could some of the rescue be due to changes in sirtuin activity, or do you predict sirtuin independent effects?
So that's a great question. With it, I believe we don't have still the numbers of the sirtuin activity analysis. We want to do that, but we indeed are interested in checking the sirtuins because we decided to utilize niacin to avoid precisely inhibiting sirtuins from the other side of the biosynthetic pathway by using nicotinamine. It could be it was another option to use as a supplementation, but we wanted to avoid product inhibition of sirtuins, and indeed they are directly correlated to mitochondrial function. But so we were interested in check on sirtuin activity. We did check on expression levels. We did not see any increase in mRNA. What we did observe, and it's where results that were not very conclusive, but in Western blotting, sirtuin-experiencing animals showed an increase in sirtuin in total amount of protein, and niacin was able to decrease this amount of sirtuin, this increase in sirtuin. But we do not have the results on the activity yet, I believe.
Great. So unfortunately, we're out of time, but I really appreciate the great talks everyone gave. Thank you, everyone. And this actually concludes day 2 of the Cachexia conference. Thank you for joining us, and we'll see you tomorrow morning at 8:30 a.m. Take care, everyone.